Welcome back to another video of the Paper 1-3 series. If you aren't a member of the DF Edu Squad, you got to join now, as membership is free and it only involves the click of the subscribe button. I will be going through the 9700 October-November 2018 Paper 1-3, and all you got to do is like, sit back and finally watch and learn. Let's then get straight to the questions. Question 1. Which statement explains why it is necessary to use an electron microscope to see the cristae of a mitochondrion? The important point over here is that the electron microscope has a higher resolution than the light microscope. Therefore, it is possible to view the intricate details of organelles such as the mitochondrion. Electron microscope has a higher resolution of 0.5 nanometers and light microscope has only 200 nanometer. Therefore, lower the number, higher the resolution. And if you go to the answers A, B, C and D, A. The magnification of the electron microscope is greater than that of the light microscope. Magnification has nothing to do with why we can see the cristae of a mitochondrion, as magnification is nothing but how much larger the image size is compared to the actual size. B. The membranes of the cristae are separated by a distance greater than 200 nanometers. If they are separated by a distance greater than 200 nanometers, that means we can view through a light microscope. However, the question is saying why it is necessary to use only an electron microscope to see the cristae. Therefore, it should be less than 200 nanometers in order to view through an electron microscope. Hence, answer B is incorrect. Answer C. The resolution of a student microscope using daylight is too low. Yes, that is correct, as light microscope has a resolution of 200 nanometers, whereas electron microscope has a resolution of 0.5 nanometers. Lower the number, that means higher the resolution. Hence, C is the correct answer. Answer D. The wavelength of an electron beam is longer than the wavelength of light. That is incorrect, as shorter the wavelength of electron beam, higher the resolution. Question 2. A prokaryotic cell which is 0.25 micrometer in diameter is magnified 50,000 times on an electron micrograph. How big will its diameter be in the electron micrograph? The question is asking about the image size, and they have given the magnification as well as the actual size. The magnification is 50,000 and the actual size is 0.25 micrometer. Since the answer is in millimeter, you first have to convert the actual size, which is in micrometer, to millimeter. And how do you do that? Well, simply you just multiply it by 1000, as 1 micrometer is equal to 1000 millimeters. And if you use the formula magnification is equal to image size upon actual size, you can find out the image size. And if you substitute in the values given, you will get the image size as 1.25 into 10 to the power 1 millimeter. Therefore, the answer is C. Question 3. When making measurements in experiments, which methods could have parallax errors? 1. Using a calibrated eyepiece graticule to measure length. 2. Using a measuring cylinder to measure volume. 3. Using a ruler to measure length of a shoot. Parallax errors is where viewing by the eye leads to error in judgment. When you view eyepiece graticule through the eyepiece lens, you are viewing at right angles, hence there is no parallax error. Whereas 2 and 3, using a measuring cylinder to measure volume, yes, it could lead to parallax errors according to where you place your eye to view the volume. 3. Using a ruler to measure length of a shoot, yes, again it could lead to parallax errors, as different positions of viewing can lead to different results. Hence the answer is D, 2 and 3 only. Question 4. The diagram is a drawing from an electron micrograph of a typical animal cell. What is the function of the membrane system labeled X? As you can see in X, the outer membrane is continuous with the nucleus. Therefore, it should be of the endoplasmic reticulum. And how do you know which endoplasmic reticulum it is? Well, since there are tiny black granules, you can identify it as the rough endoplasmic reticulum. And what does the rough endoplasmic reticulum do? Well, the rough endoplasmic reticulum is involved in protein synthesis due to ribosomes being present, as well as transport. Hence, the answer is B, protein synthesis and transport. Why not A, lipid synthesis? Well, lipid synthesis occurs in the smooth endoplasmic reticulum, not in the rough endoplasmic reticulum. And since X is labeled as rough endoplasmic reticulum, the answer is B. Question 5. The diagram is a drawing from an electron micrograph of a typical animal cell. Where would nucleic acid be found? 1 is the cytoplasm, 2 is the ribosome, 3 is the mitochondrion. Now wherever RNA is found, nucleic acid is present. and cytoplasm will have nucleic acid in the form of mRNA. Does ribosomes have nucleic acid? Yes, because ribosomes have rRNA, which is ribosomal RNA. Therefore, since RNA involves nucleic acids, nucleic acid is found in the ribosome. If you look at 3, well, 3 is a mitochondrion, and mitochondrion has 70s ribosomes. Therefore, rRNA is present. Hence, the answer is A, 1, 2 and 3. Question 6. The diagram shows some similarities between chloroplasts, mitochondria and typical prokaryotes. Which row is correct? A key thing you should remember is that all 3 of them do have 70s ribosomes and circular DNA. Therefore, if you go to the answer A, B, C and D, A is incorrect as it mentions mitochondria and prokaryotes having linear DNA. B is also incorrect as chloroplasts and prokaryotes do not have linear DNA. C is also incorrect as there is a mention of linear DNA among mitochondria and chloroplasts. D, prokaryotes and mitochondria, yes, it does have 70s ribosomes. Mitochondria and chloroplasts, yes, again they do have 70s ribosomes. Hence the answer is D. Question 7. A student carried out tests on the same volume of 4 different solutions to investigate the presence of protein, starch and reducing sugar in each. The results are shown in the table. Which conclusion can be drawn from these results? Benedict's solution is used to test for reducing sugars, Biret reagent is used to test for proteins, and iodine solution is used to test for starch. The positive test for iodine solution is blue-black, which mentions that starch is present. The positive test for Biret is purple, indicating that proteins are present. The positive test for Benedict solution, well, there is a color scale, and the color scale is from blue to green, green to yellow, yellow to orange, and finally red, where blue indicates no reducing sugars present and red the highest reducing sugar present. Hence, if you go to the answers A, B, C and D, A, solution 1 has a lower protein and lower reducing sugar content than solution 2, that is incorrect. As in solution 1 it is purple, whereas in solution 2 it is pale purple, and purple indicates the presence of proteins compared to pale purple. Hence 1 has higher protein content than solution 2, not lower. Therefore A is incorrect. B, solution 2 has less starch compared to solutions 1, 3 and 4. That is incorrect as solution 2 is the only one which has blue-black color in iodine solution. Hence solution 2 is the only one with the positive test for starch. Therefore it cannot have less starch compared to the other solutions, and therefore B is incorrect. C, solution 3 has the most protein and the least starch. That is incorrect as solution 3 has no starch at all due to the color being yellow. If you go to answer D, solution 4 has a high reducing sugar content and no starch. Yes, that is correct as in solution 4 yellow is the color given under iodine solution, hence no starch, and red is the color given under Benedict solution. Therefore highest reducing sugar content is present in solution 4. Hence the answer is D. Question 8. Which words correctly complete the description of amylose? One thing you should know about is that amylose is a polymer of alpha glucose with only 1,4 linkages and it is not branched. Hence if you go to the answers A, B, C and D, A and B are incorrect as amylose is not a monomer but a polymer. And which one is correct among C and D? Well, the correct choice is D, as if you look at answer 4 where we have to choose between disaccharide and monosaccharide, glucose is a monosaccharide, hence the answer should be D. Therefore the correct complete description is the polysaccharide amylose is a polymer made up of monomers of the monosaccharide glucose, not disaccharide. Hence the answer is D. Question 9. Which statements about glycosidic bonds are correct? 1. They occur by condensation reactions between the OH groups of 2 molecules. 2. They occur only between glucose molecules. 3. The bonds can only be formed between carbon 1 and carbon 4 or carbon 1 and carbon 6 of adjacent molecules. 4. Hydrolysis of the bonds releases energy. Now statement 2 is obviously incorrect as glycosidic bonds form between 2 sugar units, not only glucose, and one such example is fructose and glucose joining to form sucrose. Therefore, answers which have statement 2 is incorrect, and that leaves us with the answer being either B or D. Why is D incorrect? Well, if you look at statement 3, the bonds can only be formed between carbon 1 and carbon 4 or carbon 1 and carbon 6 of adjacent molecules. Well, glycosidic bonds can also form between carbon 1 and carbon 3. Hence, the answer is B, 1 and 4, as statement 2 and statement 3 are incorrect. If you look at statement 1, they occur by condensation reactions between the OH groups. Yes, that is correct, as bond formation involves condensation. 4. Hydrolysis of the bonds releases energy. That is also correct, as bond breaking involves hydrolysis, and when you break bonds, you get energy. Hence, the answer is B. Question 10. What is the general formula for glycogen? When glycogen is formed by condensation, a water molecule is removed. Hence, from C6H12O6, which is the monomer, that is glucose, you get C6H10O5 once you subtract 2 hydrogens and 1 oxygen as one water molecule leaves. And that leaves us with the answer being C, C6H10O5 times N. Question 11. Which statements are correct? 1. In a triglyceride molecule, the bonds between the fatty acids and the glycerol molecule are called ester bonds. 2. Triglycerides are formed by a condensation reaction between the hydroxyl group of a fatty acid molecule and one of the carboxyl groups of the glycerol molecule. 3. Triglycerides are insoluble in water because the fatty acid carbon chain is nonpolar. Statement 2 is incorrect, as it is the other way round. Hydroxyl group is from the glycerol molecule and carboxyl group is from the fatty acid, as hydroxyl means OH and glycerol is an alcohol. Therefore, hydroxyl is from glycerol and carboxyl, which is COOH, is from the fatty acid. Hence, statement 2 is incorrect, and that leaves us with the answer being C, 1 and 3 only. In a triglyceride molecule, the bonds between fatty acids and glycerol molecule are called ester bonds. Yes, that is correct. Triglycerides are insoluble in water because the fatty acid carbon chain is nonpolar. Yes, that is also correct, as triglycerides are insoluble in water but soluble in alcohol. Question 12. The table shows the diameter of some atoms when they form bonds. The approximate length of the amino acid shown was estimated using the figures in the table. What would be the approximate length of a dipeptide formed using this amino acid? When dipeptide bond forms, there will be totally 2 amino acids, and since this given amino acid is of length 0.7 nanometer, when 2 amino acids join together, we can assume that the total length will be 1.4 nanometers. However, when a dipeptide is formed, a water molecule is removed. Therefore, it would not be 1.4 nanometer as a water molecule is removed, but a slightly smaller length. And that leaves us with the answer being B, 1.2 nanometers, as it obviously cannot be 1.7 nanometers due to 2 amino acids being the length of 1.4 and not anything greater than 1.4. And when a water molecule is removed, it will be a little less than 1.4 nanometers, as a water molecule only involves a small diameter. Hence the answer is B, 1.2 nanometers. Question 13. Which statement about the typical modes of action of a competitive inhibitor and a non-competitive inhibitor is correct? A key thing you should remember is that competitive inhibitor does not cause any change in Vmax, whereas non-competitive inhibitor does, as it reduces the Vmax. Both do, however, cause the increase in Km as enzyme affinity towards the substrate decreases. Therefore, if you go through the answers A, B, C and D, the correct answer is D. Competitive inhibitors will not alter Vmax. Non-competitive inhibitors will reduce Vmax. Let's look at why the other answers are incorrect. A. Competitive inhibitors can bind to alternative sites. That is incorrect, as competitive inhibitors, they bind to the active site of an enzyme. Only non-competitive inhibitors can bind to alternative sites. B. Competitive inhibitors have exactly the same shape as a substrate. Yes, that is correct. Non-competitive inhibitors can have any shape. Well, although this statement can be true, this doesn't describe the typical mode of action, as the question asks about how the inhibitor is related to the typical modes of action. C. Competitive inhibitors may be used to regulate enzyme activity. Non-competitive inhibitors have no functions in enzyme regulation. That is incorrect, as both are involved in enzyme regulation, as inhibitors is a feature that affects the rate of an enzyme reaction. Question 14. Some animals produce antimicrobial proteins which protect them from pathogens. These proteins could be used to kill human pathogens. However, when used as a medicine, they are broken down by protein digesting enzymes. Replacing one of the amino acids found in the protein with one that had been synthesized in the laboratory resulted in a modified protein that was not broken down. What could explain why this modified protein was not broken down by the protein digesting enzymes? 1. The modified protein has a different tertiary structure to the original protein. 2. The modified protein is not complementary in shape to the enzyme's active site. 3. The modified protein is unable to induce a fit with the protein digesting enzyme. All proteins do have a tertiary structure, hence a specific shape. Therefore, statement 1 is obviously correct. 2. The modified protein is not complementary in shape to the enzyme's active site. Yes, as if it was complementary, then it could bind to the active site of the enzyme and can then be broken down. Hence, statement 2 is also a possible explanation. 3. The modified protein is unable to induce a fit. Yes, as again it is unable to bind with the enzyme and enzyme cannot carry out its function. Therefore, the modified protein will not be broken down by the enzyme. Hence, all 3 are possible explanations and the answer is A, 1, 2 and 3. Question 15. An investigation was carried out on the effect of temperature on an enzyme catalyzed reaction. The enzyme and its substrate were initially placed into separate test tubes and raised to the temperature required. They were then mixed and placed into 4 tubes A, B, C and D. These tubes were incubated for the time and at the temperature stated. The mass of the product formed was then measured. In which tube was the rate of reaction highest? The rate of reaction is either the time taken for the substrate to disappear or the product to be formed. Since mass of product is given, we consider the rate as mass of product formed upon the time taken. Hence, I have mentioned towards the sides the rate of each reaction, and as you can see, the highest is of B, as B has a rate of reaction of 0.167 and that is the highest compared to all the other answers. Therefore, in B, the rate of reaction is the highest. Question 16. In an experiment, the proteins in the cell surface membrane of a living cell were labeled with a fluorescent dye. A small area of the membrane was observed using a microscope. A laser was then used to permanently destroy the fluorescence of the dye molecules in this field of view. The fluorescence in this field of view was later seen to return. What does this experiment show? A. Proteins are found only on the outer surface of cell surface membranes. That is incorrect, as proteins can also be found spanning the whole cell surface membrane, known as transmembrane proteins. B. Proteins in the outer layer of a bilayer do not penetrate into the inner layer. Well, that is incorrect, as proteins are able to move about and diffuse within the phospholipid bilayer. C. Proteins move freely in the phospholipids of a bilayer. Yes, as I mentioned, proteins are able to move about due to the fluid nature of proteins as described in the fluid mosaic model. Hence, answer C is correct. D. The cell surface membrane of the cell is a bilayer. Although D is correct, it doesn't explain the results of the experiment. The result shows that although the fluorescence disappeared, after some time it was seen to return. Therefore, the only possible explanation is that proteins move freely in the phospholipids of a bilayer. Question 17. In plants adapted to cold conditions, their cell surface membranes change as the weather gets colder, allowing the plants to carry out exocytosis, which change occurs in their cell surface membranes. In cold conditions, the plants try to increase their fluidity of the membrane. Hence, the answer should consist of the mention of unsaturated phospholipids. As unsaturated phospholipids, they have double bonds in their fatty acid tails, and double bonds in fatty acid tails leads to bent tails, causing more fluidity. Therefore, the answer is D, an increase in the ratio of unsaturated phospholipids to saturated phospholipids. Why is answer A incorrect? A decrease in the ratio of proteins to saturated phospholipids? Well, that doesn't cause any change in fluidity. B. A decrease in the ratio of unsaturated phospholipids to saturated phospholipids? That is incorrect, as that will reduce the fluidity, because unsaturated phospholipids increases fluidity, and if they are reduced, then that means the fluidity decreases. C. An increase in the ratio of proteins to unsaturated phospholipids? That again has no effect. Therefore, the answer is D. Question 18. Which statement describes endocytosis? Endocytosis is a process by which a vesicle forms and uses energy to take in substances into the cell. Therefore, if you go through the answers A, B, C and D, answers A and B are incorrect, as it doesn't mention the term vesicle, as vesicle formation is important in both endocytosis and exocytosis. Therefore, considering between answer C and D, both endocytosis and exocytosis requires energy. Therefore, answer D is incorrect, and that leaves us with the answer being C, movement across a membrane into a cell using a vesicle and requiring energy. Question 19. An indicator is colorless in acid and pink in alkali. In an experiment, a petri dish of agar was prepared using an acidic solution of this indicator. A disc of agar 1 cm in diameter was removed from the center to create a well. A white card showing circular marker lines 1 cm apart was placed underneath the petri dish. 1 cm cube alkali solution was put into the well in the agar and a stopwatch was started. A circular disc of pink color appeared and spread through the agar. It reached the first marker line in a short time but took longer to reach the second marker line and a very long time to reach the third marker line. What explains these observations? This, although a very long question, if you break it down into the key points of the question, such as colorless in acid and pink in alkali, as well as the time taken for the pink color to reach the markers, it clearly shows that this process is diffusion, as it doesn't involve any transport proteins anywhere in this question. Therefore, it cannot be facilitated diffusion, as facilitated diffusion involves transport proteins. Hence, considering between answers C and D. Why is D incorrect? As it mentions simple diffusion of the indicator. However, since the pink color spread, it indicates a diffusion of alkali, as the indicator is pink in alkali. Therefore, it is a diffusion of alkali solution, not of the indicator, as the indicator could be colorless in acid as well as pink in alkali. Therefore, it is better to say simple diffusion of alkali solution, since the pink color is seen to appear and spread. Question 20. How many copies of each DNA molecule will be found in the cell at each stage of the mitotic cell cycle? G2 of interphase, metaphase, cytokinesis. G2 phase occurs after the S phase of interphase, where DNA replication occurs in the S phase. Hence, when DNA replication occurs, 2 copies of each DNA molecule will be produced. Therefore, G2 will also have 2 copies, since G2 is a phase that occurs after the S phase. Therefore, that leaves us with the answer being either C or D. Since metaphase is at the start of the mitosis, it will too have 2 copies, as no division of cell occurs until then. Cell division occurs towards the end of the mitotic cell cycle. Hence, the answer is D. G2 of interphase, 2 copies, metaphase 2 copies, cytokinesis 2 copies. Question 21. Centromere, chromatids and telomeres are parts of a chromosome in a eukaryotic cell. Which row correctly describes these structures? DNA is a double-stranded molecule. Therefore, if you see any answer having single-stranded DNA, that is incorrect. If we go to the answers A, B, C and D, both B and D has a mention of single-stranded DNA. Therefore, they are incorrect. Why is answer C incorrect? Well, they have mentioned site of production of spindle microtubules under centromere, whereas centromere is not the site of production of spindle microtubules but the site of attachment of the spindle microtubules. The production of spindle microtubules is done by the centriole. Hence, C is incorrect, and that leaves us with the answer being A. Binds the 2 sister chromatids of a chromosome together. Yes, that is correct of a centromere. Chromatid contains histone proteins and a molecule of double-stranded DNA. Yes, that is also correct. Telomere protects the tips of each chromatid from shortening at each mitotic division. Yes, because they are present to prevent the loss of genes. Question 22. Which bases found in nucleic acids are purines? The best way to remember this is a term called Gapur, where guanine and adenine are purines. Therefore, the answer is B, guanine and adenine. Question 23. A length of double-stranded DNA contains 120 nucleotides and codes for a section of a polypeptide. What is the maximum length of this section of a polypeptide? When a double-stranded DNA is forming a polypeptide, it should be transcribed and translated. During the process of transcription, the double-stranded DNA unwinds or unzips, forming 2 separate strands, and the transcription occurs only on 1 strand. Therefore, since they have given 120 nucleotides for double-stranded DNA, we have to consider for 1 strand, that is 120 nucleotides divided by 2. That will give you 60 nucleotides on 1 strand. And since 3 nucleotides codes for 1 amino acid, that will give us 60 divided by 3, that is 20 amino acids. Hence, the maximum length of this section of a polypeptide is 20 amino acids, and the answer is A. Question 24. 4 students were asked to suggest a set of 4 labels to add to a plan diagram of a transverse section of a dicotyledonous leaf. Which student suggested a correct set of labels? The key part of this question is plan diagram. In a plan diagram, it only involves the labeling of separate layers and not individual cells. Therefore, answers B, C and D are incorrect, as D mentions companion cells, and cells again are not mentioned in a plan diagram. C involves xylem vessel elements, where xylem vessel elements are the cells that form the tissue xylem. Since they have mentioned xylem vessel elements, and xylem vessel elements are cells. You cannot add that label to a plant diagram. B. B also has xylem vessel elements, hence it is incorrect. Therefore, the correct answer is A. Lower epidermis, yes, that is a layer. Phloem, that is a tissue. Spongy mesophil, that is again one layer of the transverse section of a leaf. Palisade mesophil, that is again another layer. Hence the answer is A. Question 25. Photomicrograph X shows a transverse section to a typical stem. Photomicrograph Y shows a longitudinal section to one type of cell shown in photomicrograph X. Which letter represents cells from photomicrograph Y? The photomicrograph Y clearly shows the presence of sieve plates or end balls, and presence of end balls or sieve plates are only in phloem. Therefore, it should be represented by C in photomicrograph X, as C represents phloem, whereas the other answers A, B and D do not. Question 26. By which process could water rise up through xylem vessels in a plant root when the shoot has been removed? When the shoot has been removed, there would be no leaves, hence there is no transpiration pull, and that leaves us with the answer being either B or D. Water travels across xylem due to cohesive and adhesive forces. Therefore, capillarity is involved. Hence, the answer is B. Question 27. Which changes occur to the water potential and the volume of liquid in the phloem sieve tube element when carbohydrate is moved into a sink? This describes the unloading of sucrose. As the carbohydrate, it leaves the phloem sieve tube element and enters the sink, and that leads to water potential in the phloem sieve tube increasing, and that causes the volume to decrease as water leaves and flows to regions of lower water potential. Therefore, the answer is C. Water potential of the sieve tube element raises, hence that leads to volume decreasing as the water will flow from the higher water potential to regions of lower water potential, leading to a decrease in volume. Question 28. The graph shows a diameter of a tree trunk over time. Which statement explains these changes? In these kind of questions where diameter of tree trunk is involved, you have to look out for the word tension forces in the answers. Therefore, if you go through the answers A, B, C and D, only answer A involves tension forces. Hence, the correct answer is A. As when the tree trunk is under tension, the diameter will decrease. Hence, the answer is A. Question 29. What is found in all blood vessels, lymph and tissue fluid? 1. Carbon dioxide, 2. Glucose, 3. White blood cells, 4. Antibodies. Generally, all are found in blood vessels, lymph and tissue fluid. Therefore, the answer is A. 1, 2, 3 and 4. Only large plasma proteins are not found in tissue fluid. Since they haven't mentioned anywhere of large plasma proteins, the answer is A. Carbon dioxide, glucose, white blood cells and antibodies are found in all three of them. Question 30. Which reactions will be taking place in blood that is passing through active tissues? Reaction 3 is obviously incorrect, as carbonic acid formation is only involved with water and carbon dioxide in the presence of carbonic anhydrase, which is an enzyme that catalyzes the reaction between water and carbon dioxide. Therefore, 3 is incorrect, as no other reaction leads to carbonic acid formation. And since A, C and D has 3 in it, the correct answer is B, 1, 2 and 4 only. Question 31. The graph shows a percentage saturation of hemoglobin with oxygen at a range of partial pressures of oxygen and at 2 partial pressures of carbon dioxide. One curve is at the partial pressure of carbon dioxide in the lungs and the other curve at the partial pressure of carbon dioxide in the liver. Which points on the curves correspond to the percentage saturation of hemoglobin at the lungs and at the liver? Now remember, only one curve belongs to the lungs and the other to the liver. In the lungs, obviously there will be high percentage saturation of hemoglobin with oxygen and the highest out of all the points given. Hence, it has to be L. Therefore, we have to consider between A and B, as L corresponds to the percentage saturation of hemoglobin at the lungs. And now looking at the liver. Well, in the liver there will be higher partial pressure of carbon dioxide and the curve shifts to the right only when there is higher partial pressure of carbon dioxide. And since point L is on the curve to the left belongs to the lungs and curve to the right belongs to the liver. Therefore, the answer cannot be obviously A, as L and J are both present on the same curve. However, they are asking points that correspond to the saturation at the lungs and at the liver. Therefore, the points should be on two different curves. And that leaves us with the answer being B, L and K. Question 32. A person moves from sea level to live at a high altitude. After 3 months living at high altitude, which change would be found in this person when at rest? Living at high altitude causes the red blood cell count to increase in order to compensate for the reduced partial pressures of oxygen. Therefore, if you go to the answers A, B, C and D, the best possible choice is answer D, increased mass of red blood cells. As more the red blood cells, more oxygen will be transported around the body. Therefore, the answer is D. Question 33. The diagram shows 3 of the features found in tissues of the gas exchange system. Which structures of the gas exchange system could be represented at position X in the diagram? 1 bronchiole, 2 trachea, 3 bronchus. Now X is at the center of all the 3 circles. That means X has all 3: ciliated epithelium, goblet cells and cartilage. Therefore, X cannot be 1, which is bronchiole, as bronchioles do not have goblet cells. And since answers A, B and C has 1 in it, all 3 of them are wrong, leaving us with the answer being D, 2 and 3 only. Trachea and bronchus, yes, they do have all 3: ciliated epithelium, goblet cells as well as cartilage. The only difference is, in the trachea, the cartilage is in the form of C-shaped rings. In the bronchus, the cartilage is in the form of irregular blocks or irregular plates. Question 34. Chronic obstructive pulmonary disease includes bronchitis and emphysema. Which row shows the effects of bronchitis? Now I have underlined which condition they are looking at. They are looking at bronchitis. Therefore, infection is definitely present, as the cilia would be paralyzed, and when cilia is paralyzed, the mucus accumulates along with trapped pathogens leading to infection. Hence, we have to consider between the answer being B or C. T helper lymphocytes also will increase as a result when it comes across the trapped pathogens, as the trapped pathogens could lead to an immune response. An immune response means T helper cells will increase. Therefore, the answer is C. T helper cells increased. Bronchioles are scarred. Yes, because formation of scarred tissue. Infection is present. Yes, that is also correct. Hence, the answer is C. Question 35. Which factors affect the distribution of the disease malaria? Distribution of the disease has to do with the mosquitoes. Hence, their habitat is of main importance, along with migration of people from regions where there is high incidence of malaria. Therefore, if you go to the answers, draining swarms, yes, that does affect the distribution of the disease malaria as it affects their habitat. 2. Vaccination. No, vaccination is not going to affect the distribution of disease malaria. 3. Antibiotics. That again won't affect the distribution as it doesn't have anything to do with the habitat as well as the disease causing agent, which is a protoctist, as antibiotics only work on bacteria. 4. Migration of people due to conflict. Yes, as migration could lead to regions where there is no cases of malaria having some cases of malaria due to people entering from countries where there is high incidence. 5. Climate. Yes, again climate does affect the habitat of the Anopheles mosquitoes. Therefore, the answer is 1, 4 and 5, and that is B. Question 36. Which diseases cannot be treated with antibiotics? 1. Cholera. 2. HIV/AIDS. 3. Measles. The diseases that cannot be treated with antibiotics are viruses, as viruses are unaffected by antibiotics. Hence, the answer is 2 and 3 only, as 1 is caused by a bacterium known as Vibrio chloriae. Therefore, 1 can be treated with antibiotics. Since the question is asking which cannot be treated, the answer is 2 and 3, as both 2 and 3 are caused by viruses. Question 37. A West African country introduced a measles vaccination during a measles epidemic. Later, it was realized that vaccinated children were more likely to survive childhood than unvaccinated children, even when there were no measles epidemics. The vaccine had given the children some protection against other pathogenic infections. Which statement could account for this extra protection? A. B lymphocytes produced memory cells which gave the children passive immunity to these infections. That is incorrect, as when memory cells are produced, that leads to active immunity, as active immunity only has to do with memory cells being produced. Passive immunity doesn't have anything to do with memory cells being produced. Hence, answer A is incorrect. B. Memory cells produce plasma cells which secreted anti-measles antibodies that bound to antigens that closely resemble measles antigens. Yes, there is a possible statement, as antibodies are specific in nature and they target only specific antigens or closely related antigens. As closely related antigens could have similar shapes to the actual antigen, therefore the anti-measles antibodies could attach to these closely resembling measles antigens. Therefore, answer B is the correct choice. C. Memory cells produce plasma cells which secreted anti-measles antibodies that bound to any antigen. That is incorrect, as anti-measles antibodies cannot bind to any antigen. They only bind to antigens which are specific in nature. D. T lymphocytes produce memory cells which gave the children natural immunity against these other infections. T lymphocytes do not produce antibodies, therefore reduced immunity and reduced protection against other pathogenic infections. Therefore, D is also incorrect. And that leaves us with the answer being B. Question 38. Which graph shows the changes in antibody concentration following a second exposure to the same infection? If the person is exposed to the same infection for the second time, then there will be a rapid rise in antibody concentrations when exposed to the second time. Therefore, the only answer with rapid rise in antibody concentration over time is A. If you look at B, there is no rapid rise, as both do have similar looking concentrations. C. After the second exposure, there is less antibody concentrations than the first exposure, which cannot be true. Therefore, C is incorrect. And at D, well, after the second exposure, there seems to be very little antibody concentrations. Hence, D is also incorrect. Question 39. Where are antigens found? On surface of pathogen, in blood plasma. Antigens are definitely found on the surface of pathogen and are not found in blood, but rather bounded to a molecule. Therefore, the answer is B. On surface of pathogen, yes, they are found. In blood plasma, no, they are not found. Question 40. Whooping cough is a highly infectious disease of the gas exchange system caused by the bacterium. Which method provides protection to infants against whooping cough and reduces the chance of developing this disease later in life? A short course of more than one type of antibiotic. That is incorrect, as antibiotics should not be used as preventative medicine but rather as a cure. Therefore, antibiotics are only used if the infant develops whooping cough, not to prevent them developing this disease later in life. Therefore, A is incorrect. B. A 6-month course of only one type of antibiotic. That is again incorrect, as antibiotics should not be used as preventative medicine. C. Injections of antibodies specific to the bacterium. That is also incorrect, as injections of antibody leads to passive immunity, not active immunity. Therefore, if passive immunity is involved, then there is no chance of developing memory cells, hence increased chances of developing this disease later in life. D. Injections of dead bacteria. Yes, because when you inject dead bacteria, it leads to an immune response. An immune response causes memory cells to be produced and therefore active immunity. And when active immunity is produced, that reduces the chance of developing the disease later in life. Therefore, the answer is D. There goes our 40 questions. Hope you found this video useful. I can only be sure if you do give this video a like. Let me know in the comment section below any clarifications or any sort of paper wanted to be done, and I will hop on it. Thank you for sticking by and stay tuned for more.